This is the second video of chapter 9, and at the end of it, the student will be able to define the resting potential of a neuron, give the origin of the resting potential, and finally to describe how the resting potential is maintained. First, if we insert the microelectrode of a voltmeter into the axon of a neuron and we keep the other electrode outside the axon, the voltmeter records a certain negative value of potential or voltage. So, what's the value of this voltage? What's its origin? Is it constant or variable? Let's discover the answer. First, we will determine the value of resting potential. We use an oscilloscope and a giant axon of a squid put in sea water. The microelectrode of the oscilloscope is inserted into the axon. The other electrode is kept outside the axon. The oscilloscope records a value of -70 millivolt, considered as resting potential of the neuron. What does this value mean? It means that the intracellular medium of the cell at rest is 70 millivolt lower than the extracellular medium. As we saw, the value is negative. But why? To find the answer, let's examine this table that shows the concentrations of different ions in the intra and extracellular media of marine animals represented by 1 and mammals represented by 2. What can we draw out from this table? We can see that the concentrations of Na plus and Cl minus in the intracellular medium are less than those in the extracellular medium. In the contrary, the concentration of K plus in the intracellular medium is greater than that in the extracellular medium. Plus, there are proteins and nucleic acids, DNA and RNA, that are negatively charged in the intracellular medium. Overall, there is an unequal distribution of ions between the two media. This leads to the presence of a negative charge in the intracellular medium and a positive charge in the extracellular medium. And now, how the resting potential is maintained. This is the cell membrane of the neuron. This is the extracellular medium, positively charged. Concentration of Na plus and Cl minus in this extracellular medium are greater than those in the intracellular medium, the cytoplasm, which is negatively charged, and in which there are the A minus. A minus represents the proteins and the nucleic acids negatively charged. And as you see, the concentration of K plus in this medium is greater than that in the extracellular medium. In fact, the resting potential is maintained by a double transportation of ions. Let's see the first one. These are the ionic channels through which ions can diffuse from a medium to another. The diffusion through ionic channels is done according to the concentration gradient, which means that the ions pass from the medium of high concentration to the medium of low concentration. And according to this gradient, K plus ions leave the cell while Na plus ions enter it. But the quantity of K plus that leaves is much greater than that of Na plus that enters. This means that the amount of positive charge leaving is much greater than that entering. This helps in the maintenance of the negative charge in the intracellular medium. To be noticed that diffusion is said to be passive transportation of ions. Why? Because it doesn't require energy and it occurs due to the concentration gradient. Usually, diffusion stops when the concentrations of the ion in the intra and extracellular media become equal. Do we arrive at such moment? Actually, no, because there is a pump called Na plus K plus ATPase pump, or simply Na plus K plus pump. What happens at the level of this pump? Look at this. The K plus that leaves the cell through the ionic channel re-enters by the pump, and the Na plus that enters the neuron through the channels leave by the pump. So the role of this pump is to restore the initial concentrations of each ion and to keep the unequal distribution of ions between extra and intracellular media. And since the pump consumes energy in the form of ATP during the transportation of ion counter to the concentration gradient, this kind of transportation is said to be active.